Okay, let's get started. Hi, everybody. My name is Eric Dufresne. I'm a professor at ETH. I've been there for about 2.5 years or so. Before that, I spent the rest of my life in the US, as you can tell by the way I talk. I was a professor at Yale for about 12 years before I moved to ETH. I'm the only lecturer who's giving, I guess, several lectures, let's say, who's an experimentalist. So hopefully my style will be refreshingly different and not frustratingly different. We'll see as we go along. My laboratory is called the Laboratory of Soft and Living Materials, and our lab works sort of is split in half, you could say, and about half of us, when we say work with living materials, we mean we grow cells in the lab and we do measurements on cells growing in the lab, looking at them with microscopes. And then we also look at synthetic systems, typically polymeric systems these days, and liquids and their interfaces. And one of the things I really try to do in my lab is I call it biologically inspired physics. So when you look at a living system, it always generates lots of different hypotheses of what's going on. And it's often very difficult to test that inside of a cell or an organism. Sometimes it's even hard to test it in vitro with purified proteins. We often, make a hypothesis about something that's happening in a cell and then just do it in a system that's completely different from synthetic polymers to try to understand what's happening. And so all the work that my lab has done in the active matter world has been so far that we've published has been in the context of living systems. So, and today in my first lecture, I want to give you an overview of some of the phenomenology of living systems and how we can think about them as interesting examples of active matter. And also right now in my lab, we're trying to develop new model systems, model synthetic systems for probing active matter that I'll be happy to tell you about over a break. So since I'm a bit of an outsider and a bit of an experimentalist, I wanted to start really, really broad. And I want to just start by asking the question, what do we really mean about active matter? And I feel like we all know what it is when we see it. But I just want to force ourselves to be a little bit more careful. And I'm proposing definitions that I'm making up myself, so they may not be right. So I'd be happy to receive any feedback that you have about them. And so, the way I think about active systems is that they're systems and which are active. And what I mean by active specifically is that they're active when they're driven out of equilibrium in a special way. So we can drive, as Ludovic said in his last lecture, we can drive a system out of equilibrium by injecting energy at the boundaries, by shearing or shaking, right? But the thing, so we inject energy macroscopically, right? But the things when I think of an active system, I always think it's a system driven out of equilibrium by microscopic irreversible processes in the bulk. Okay. So there are things happening in the material at the microscopic level, perhaps probably at the molecular level, that produce entropy. So one picture I really like to think about that's quite general, to think about the microscopic processes of active matter is to think about cyclic processes, right? And so recently McIntosh and a bunch of other people had a nice paper where they were arguing that really the thing that makes something active is the breaking of detailed balance at the microscopic level. And so and they argued that the key thing to think about is looking for cycles in phase space. And so imagine that if you have some I won't say a microstate technically as in statistical mechanics, but some microscopic state of the system that is driven in a loop. It might not be driven deterministically in a loop. Usually there's some reverse process, right, that happens with some low probability. And the idea is that in active systems, you really break detail balance at the microscopic level. That's a picture I like a lot. Okay, so active systems can exist in all different states, and my interest is really in what I would call active materials, and I want to define what I mean by an active material. The first thing about, there's two things you want to make an active material. You want homogeneity in space, in time and in space, okay? Otherwise, it's really hard to think about something as a material if you can't really pin it down, its properties. And so first, I just want to talk about, talk about homogeneity in time, or a steady state. So active systems can be in steady state. And they can reach a macroscopic steady state. They can do it, but in order to do it, they need to exchange energy and or matter with the surroundings. Okay, so here, what's interesting is I proposed a real microscopic molecular picture for how to think of what active systems are. Here I'm sort of jumping to a macroscopic picture, and it's a really old picture. If I draw my system as a black box, or here as a green box, right, with a white border, if I wanted to think about what my system looks like, it would look like this. So there's a mass flow in, which I would call a fuel. There's a mass flow out, which I would call the waste, and then it might do some work on its surroundings and maybe reject some heat. Okay. When you draw an active system in that really schematic way, it reminds us of a very important fact, is that there's nothing new about active systems. This is the picture nothing new, no, there's lots of new physics to discover, but people have been thinking about this for a long time. This is a sort of diagram someone would use to describe schematic diagram that you would use to describe some sort of heat engine or any other machine that can use this fuel to do work. So, right, so let's not think, let's not force ourselves to, my computer wants to update. No, do not update. It always happens at the worst time. Let's remember active matter is really big. Active systems are huge. It's not just swimming particles. It's also engines and all sorts of things. It's a really big area. It's also true of living organisms. So of course, living organisms don't truly reach a steady state because we don't live forever, right? But over some reasonable time scale, steady state is an excellent approximation for what an organism does, okay? At that level. Okay, and now I want to talk about homogeneity in space and really introduce this concept of active materials. And this is, for me, I'm trained as a physicist, but at ETH, I'm in a material science department. For me, this is really a driving concept. I want to make active materials, and they are, there aren't that many examples. I think there might be 0 examples of engineered ones or synthetic ones. But the idea of what I hope they would be is they're active systems with well-defined continuum scale properties. Right? A lot of times in active matter, we really focus on sort of how individual things swim, or how correlations between maybe small numbers of swimmers. But as we progress in active matter, as we heard today from Ludovic, now we're really trying to, the next step is to really think about things at the continuum scale, right? How does activity change the properties of a material? And I'll give you some examples of this. And again, this is biologically motivated because there are all sorts, I'll give you lots of examples in a moment, but I'll just give you one. Well, I'll just say tissues are one class of examples, and muscle is the canonical active material, right? So what's interesting about muscle, right? It has all the normal material properties, mechanical properties that a solid would have. It has a modulus, it has a shear modulus, it has speed of sound, all those great things. But it also has a special property, which is how much stress it can generate, right? And that's a special property due to its activity. And it'd be great if we could engineer materials that have similar in that the idea of they have new emergent properties that passive materials do not have. Okay, so that's sort of setting the context a little bit and sort of defining this very important objective for me is sort of active materials. It's something I'm targeting. Now, as I mentioned earlier, all the work I've done in active systems so far has all focused on living systems. And so I just want to give you a super quick overview. Well, it depends how many questions you ask. It could be a long overview. I'm happy to take questions at any time about anything. But life is really, I like to say life is a hierarchical web of active systems, right? Life exists across scales, and all the components are deeply interconnected. And so, there are all sorts of living systems that we can think of, right? So here's a cell. It's got a length scale of about 10 to the minus 5 meters. This thing takes in nutrients. It rejects heat. It gets rid of fuel. An animal, right, is also an active system. It meets all the requirements. It's reasonably in steady state. And it has a length scale of about 10 centimeters. And at some level, you can think of the whole ecosystem of our planet as an active system. It's driven by an energy flux from the outside, the sun, and we reject heat to the universe, right, by radiation. And there's all sorts of crazy things that happen. And unfortunately, our ecosystem is not quite in steady state anymore. Hopefully it's not as bad as I fear it might be. But anyway, so of course, so there's sort of all ways to look, active matters everywhere. Let's not forget that, okay? But, many of these systems, these complex active systems, they're built from passive conventional materials in equilibrium or near equilibrium, but they can also be built out of active materials. So I want to give you some examples of active materials that you encounter when you look at biological systems to give you a sense of the diversity of their structure, both in terms of the way they're ordered and also at the length scale that they live in, and also give you a sense of what interesting properties they have that are sort of somewhat unusual. So the first one I want to tell you about is the cytoskeleton. And it's special to me at Les Houches because this is, I was a student at Les Houches like in 2004, and I went to a school about the cytoskeleton. But the point is that the cytoskeleton is a polymer network inside of most cells of your body. It can be a completely disordered polymer network, or it can have liquid crystalline order, which I think you might have heard of with Joanny, perhaps, earlier in the school. And here I'm drawing a particular type of network, a branched actin network. That's a polarized actin network. It's cross-linked. and polarized. This sort of actin network I'm drawing here with all the branches sort of pointing in the same way is characteristic of what you see right underneath this right behind a crawling cell's membrane. And it has a characteristic length scale of about 100 nanometers, a characteristic structural length scale of around 100 nanometers. Now, the cytoskeleton has all sorts of important functions inside of the cell, all of which are mechanical. Not all of which, actually. Most of them are mechanical. And some of them are classical things that non-living systems do, like they provide elasticity. The cytoskeletons can provide elasticity just because it's a cross-linked network that spans space. It has a shear modulus. Slightly more sophisticated thing they can do is they can be responsible for transport. Right, so when you have a network of actin, as I'm showing here, and it's contracting in one place and not contracting in the other, there can be a flow of fluid from one side of the cell to the other. So that's one way of driving mixing inside of a cell. Other types of cytoskeletal elements like microtubules act as little railroad tracks that can more specifically transport cargoes from one spot in a cell to another spot in a cell. And as I also mentioned, quite importantly is they can contract. So not only can they passively respond to outside stresses applied to them, but they can generate their own stress. Just like your muscle can generate stress, even this little fancy polymer network can do the same thing. Okay? Any questions about the cytoskeleton or comments about the cytoskeleton? The more questions you ask, the more likely we all are to be awake, including me. Yeah. Yeah. Yeah.
All your some requirements would be fulfilled, right? It would be non-equilibrium.
Yes.
Consume energy.
Yes.
Blah blah blah. So, but what you would probably not really want to call active matter.
Yeah. So this is where I was trying to say that it's driven by the irreversible processes in the bulk. So I guess when you drive it at the boundaries, ultimately there are microscopic irreversible processes that could generate the heat. But I think here the idea is that it's driven at the microscale. I'm not sure if that's a rigorous way of viewing it, but that's at least how I've conceived of it. Okay, so let's move on to some more examples, and we're going to sort of try to move up length scales. The next example I want to give you is a really recent example, and it's not really even clear how much the activity of what I'm about to tell you about is important. It seems at least passively, tangentially important. So there's these objects called membraneless organelles. The basic idea is they're little micron-sized blobs that live inside of cells, and they have structure at the nanometer scale. And they can be fluid, or they can be glassy. And whether or not they're fluid or glassy seems to depend. It seems to depend on how much ATP is around. So the properties of these little organelles, they're basically regions inside of the cytoplasm where certain molecules are enriched compared to others. It's a form of phase separation. Okay? Whether or not the activity is coupled to the phase separation is a really interesting question that people like Frank Eulischer are probing. As far as we know, though, so far, in terms of the function of these things, when you talk to a biologist, the main thing that they think of these things doing is basically organizing the cytoplasm. So they play a role called partitioning. So the idea is that you can basically sequester things that react inside of these membraneless organelles, and then when you need them, you can dissolve them. And whether or not activity plays an important role in the dissolution and the partitioning, I would say, is an open question, but I suspect that it does. Okay. So yeah, moving up more in scale, right? So this is basically this thing has sort of a liquid-like structure. So I also want you to appreciate the diversity of the structure in these things. The next thing I want to mention is sort of skeletal muscle cells. Okay, so they sort of look like this. Let me get my picture right here. They have stripes on them. The stripes correspond to different structures of actin and myosin. That detail is not really important here, but this periodicity is on the scale of a few microns, and the key thing about this system, instead of being disordered, is it's actually crystalline in one dimension. It has long-range translational order. Maybe not technically long-range, but for a while, there's a pretty well-defined spacing between these things. And here, of course, the key function of the system is to generate force and to contract. And there, the activity is known to be primarily responsible for liberating the free energy to do the work on the outside world. And the last, the biggest length scale I want to talk about for today is cell monolayers, and in particular, I want to talk about the endothelium briefly. So the endothelium is a two-dimensional or pseudo-two-dimensional cellular system. It consists of a cell monolayer. Okay, and it's hard, as Ludovic has already said, to draw things that are disordered quickly. But anyway, it can be disordered. The typical spacing here would be about 100 microns, corresponding to the size of a few cells. And so this is a monolayer of cells. And it does some amazing things inside of all of your blood vessels. So every blood vessel it's a tube through which the blood is flowing. This layer of cells does a couple of important things. It is a mechanical sensor. And people still argue tremendously about how this works, but the data is completely conclusive that this layer of cells is able to detect the shear stress on its surface, even though the shear stress is only on the order of 1 Pascal. So they can detect these incredibly low shear stresses, and then they transduce that information, and they basically regulate the diameter of they tell muscles around them to contract, to either contract or release the contraction to maintain a constant shear stress on the inner surface of all your blood vessels. So these are amazing things. They also have what's called a barrier function, right? So they have an important job to keep the bad cells that your body makes, cancer cells, from getting into the bloodstream and circulating. And their barrier function is also really important to make the bad cells that are from the outside from going into the rest of your body, right? So the barrier function and the mechanical sensor aspects of the endothelium are really important. And certainly, definitely the mechanical sensing part of this definitely requires active processes. There's also some really interesting physics that is not really actually I haven't seen it explored in the physics community, but there's basically an isotropic to pneumatic transition in the endothelium that's driven by shear. So if you culture cells in a dish at high density, they originally start off completely disordered, but then when you drive a shear flow over them, they sense the shear flow, they change their aspect ratio, and they align relative to the flow. So there's all sorts of really cool physics of the endothelium, and a lot of it's active. Okay, so any comments or questions about this sort of a little taste of what active systems can do in living matter? Yeah. So that is so you can probably guess what people say. So the molecular biologists all think that there is one magic molecule that senses the shear, right? And the The cell biologists, you know, are more likely to think that it has to do with a regulatory network, so that there's some cooperative effect that's sort of where there's amplification by the sort of the pathways in the cell that process information. And I had a colleague at Yale who we used to debate whether or not we thought there was sort of, you know, the interactions between cells were important. There's data, I mean, none of this is published, this is just sort of, you know, just talking and doing little experiments, but there was some preliminary suggestions that individual cells were not able to align with the shear flow, but only groups of them can. So there's definitely, I mean, and probably the answer is multi-scale, right? I mean, you know, you need some molecule on the surface to change its conformation, and then there's some amplification probably in the cytoplasm, and there's some collective coupling. I think they're all going to be needed to figure out the story. Yes? For the endothelium, I could try to track one down, and if I don't do it, find me. It'll end up in the published lecture notes if I can find one. But I don't know of a good example of the endothelial. Actually, for the alignment, I don't have a good reference, but for the aspect of the fact of it being a mechanical sensor and how the blood vessels expand and contract to maintain a constant shear stress, that's really well documented. And my former colleague from Yale, Jay Humphrey, has a really nice review article about the mechanical sensing of the endothelium and how it couples to blood vessels. And if you have difficulty finding it, find me, and we'll find it together. But I'll try to put it in my lecture notes. It's more fun to discuss. Do you really want me to keep going, or do you want to talk? Okay, I'll keep talking. All right, so I have a question for you now. Right, so what do biologists call activity? Students, please. The most important part about collaborating with biologists and doing biophysics and biologically inspired physics is just understanding what the two groups of people are saying. So what does a biologist call? Yeah. Oh, that's one very important piece of the activity. And often we reduce it to the only thing, but there's a bigger word. Literally, well, is it literally bigger? I don't know if it's literally bigger, but there's a word that encompasses a broader range of things. Ah, that is not overlapping, but bigger, involves more reactions. Larger scale, broader, it's a real big catchphrase. Yes, who said that? Yay, all right, Ramin, all right. I said already, but the students didn't say anything. Yeah, exactly. So, okay, so what is metabolism? So I'm gonna talk a lot about metabolism in this lecture and the next lecture and maybe the next two lectures depending on the speed and how many questions you ask. Yeah, we'll be talking a lot about metabolism. Right now, I wanna tell you, sort of summarize what biologists think about metabolism. And then as we go forward, I'm gonna talk to you about like what's the right way to think about it physically and how can we measure it in the lab, okay? So metabolism, it's actually really simple, but because it's so simple, it's so broad, right? It's the set of chemical processes that underlie another biology word, homeostasis, okay? And can anyone tell me what homeostasis is? Sorry, who said that? Yeah, that's a great example, right? So the key idea behind that is steady state, right? So the key thing is that hopefully, so it's the non-equilibrium steady state of life, okay? So steady state is not necessarily good for a living organism. So I used to teach at Yale, and right around the corner from Onsager and Kirkwood in the cemetery is Josiah Willard Gibbs. And I used to teach thermodynamics to engineers, and for the last lecture before the exam, I would take them to the cemetery. And I would tell them that this was a very holy place, because this is the place where the person who first described chemical equilibrium attained it himself, right? So that's a type of steady state that you don't want, right? Put the thing in the box, let it go forever, see what happens. That's equilibrium. That's not the steady state we want. Homeostasis is the non-equilibrium steady state that we enjoy, okay? So biologically, metabolism has a lot of functions, okay? So, sort of one of the most famous, simplest examples is synthesis. Okay, what we mean by synthesis is it's the chemistry of the organism. It's how it takes in elements from its environment to build the molecules that it wants. It's about building molecules. So, for example, the creation of sugar by a plant undergoing photosynthesis is an example of synthetic metabolism. Another example of something that is important metabolically is assembly. Okay, so cells and living organisms not only use energy, as we'll see in a minute, to make things, they also use it to assemble the things that they've made, and that could and there are lots of sort of really nice examples. Let's just say there's lots and lots of examples, but in the cytoskeleton, you already heard some examples of how the metabolic activity of molecular motors and the hydrolysis of ATP can change the structure of a polymeric system. And of course, also, the metabolism can make things move, right? It can, by the hydrolysis of ATP, can make a single molecular motor, and I'll talk a lot about myosin as an example today. It can make a single molecular motor move, but it's also making me move and gesticulate wildly in front of you. So important thing here is that this is really at the molecular scale, but the processes of assembly and motility really occur across all the scales of biology, okay? And biological organisms use energy to assemble and move things around. Yeah, so I like to call these three things together the physical layer, of metabolism, and there's another layer, though, that I'm going to tell you about in a second. But the important thing is all the synthetic active systems we study so far are limited to the physical layer. Right, in the active systems experimentalists have developed, right, they consume energy to synthesize things or assemble things or move things around. But the really crazy thing, which is what I call the information layer. And the information layer itself has many layers, and it's been studied like crazy by biologists. It's been studied a lot by systems biologists. I think I argue that the coupling between the information layer and the physical layer is probably, 30 years from now will be an excellent topic for a school at Les Houches. But let me tell you a little bit about the information layer in terms of some functions. So one important thing that the information layer does is it transduces things. Okay, so it transduces signals. So back to that specific example over there about the endothelium, it can measure the shear stress, right? There are molecules at the surface, right, that help measure a shear stress, and they have some physical response. Maybe they change their conformation. But then what the cells do, which is incredible, they convert that change in conformation to information. Okay, so that the change in conformation of that initial stress, directly stress sensitive molecule will then interact with other molecules inside of the cell that pass along the information that molecule on the surface has been stretched. And when you go to biology talks and they show lots of molecules with like 3 or 4 letter names and numbers, and they have lots of arrows pointing everywhere, they're usually talking about transduction, processing, I'm going to put them all together, processing and transmitting information. Okay, so this is, this altogether in the language of biology is called regulatory networks. So at least in the context of inside of a cell, it's the regulatory networks, right? So there's stuff at the surface of a cell that creates a signal when the shear stress is, you know, too high or too low, or maybe even when it's the right value. There are networks of chemical reactions in the cell that maybe integrate that signal from many spots. And then if a threshold, a nonlinear threshold is reached, that it makes a decision to do something, then that information is transmitted and then also transduced again into a physical stimulus in the real world, either as a shape change or something else. Okay, so these are regulatory. These are really the brains of the cell. Okay, the other thing that we use metabolism for in our bodies is to store information and to copy information, right? And that's primarily all about the genetic layer. And in all of these pieces, right? These are not all of these processes are processes that that are non-equilibrium at the microscopic scale. Okay, and I think, as I mentioned before, all of our work experimentally has been here in synthetic systems. I hope one day to connect these two. Anyway, that's biology in a nutshell. Any questions about or comments about? Not at all. Okay. All right, forge ahead then. Okay, so that's the end of the sort of the really touchy-feely part of my lecture today. And now for the rest of the lecture, I want to go in and dive in a little bit deeper and talk about data. Okay, and I'm going to do an experiment. I like to do experiments, but I'm going to do an experiment not in my lecture, but I'm going to do an experiment with my lecture. And I'm going to try to show a lot of data quantitatively only by drawing things on the board. Okay, so no PowerPoint allowed. I think in my next lecture I might give in and show one plot in PowerPoint because the information density is just way too high. But I want to go back to simple things now. I told you about some really complicated stuff that's far away from what we normally think about in physics first. Okay, so I want to talk about the connection, and this is one that perhaps, well, we've heard a lot about, activity and Brownian motion. And I think it's going to be, you've been hearing about this over and over again, I'm sure, for the last week or a couple weeks. Hopefully it's going to be quite different for you, because in this case, I'm not talking about swimmers in a passive medium. I'm going to talk about passive particles in an active medium. So we'll just turn things around a little bit, okay? And that's why I'm calling it Brownian motion. So to start off with, let's do some biology and talk about Robert Brown, because we have to, the Brown and Brownian motion in 1828. So if you haven't read Brown's original 1828 paper, I really highly recommend it. Just like, and this is not like brilliant people like Don telling you to go read Gibbs, right? Because you try to read Gibbs, you can't understand it at all. Like only people like Don can understand Gibbs. One day, maybe when I retire, I might understand something. But Brown's paper, it's an experimentalist paper. It's so simple, and it really shows the power of experiments so elegantly. It's easy to read. It literally, the paper starts off like, it was a hot day in Edinburgh in August, and I decided to blah, blah, blah. Anyway, it's really fun. And so he was a plant biologist, and he was interested in how the pollen gets from the male part of the plant to the female part, okay? And people had known since the invention of the microscope that sperm existed and they have tails and they're swimmers, right? But he wanted to see how, pollen got around, and the things he looked at in the microscope. I will always try to have a scale bar. If I don't put a scale bar on my sketches, then shout me out on it. He looked at microscopic objects in a light microscope, and he noticed that, they had these crazy erratic trajectories, which we all know about. Now, the most important thing is he went into it thinking that this motion was, he called it a vital motion, right? He thought it was active motion at first, but he was a good experiment. And he didn't just do the experiment with live pollen, and this is why the paper's really fun to read. He did it with dead pollen. And the best part is he tried like all sorts of different ways of killing the pollen. So first he would use some sort of poison like mercury, and the pollen particles still swam. And then he would put it in an organic solvent, and it still swam. And then he burned the pollen, and it still swam. And then eventually he comes to the conclusion that it has nothing to do with life at all, and he did the most important experiment. He grounded up some minerals and made a really, really fine powder, and he concluded by seeing the fact that even completely dead, inert things that he knew to be inert also did this. And so he reached the very, very, very important conclusion that Brownian motion is not active. He would say vital, we say active, okay? So that was the super important conclusion. All right, and you all know the story, about what happened next, right? Between roughly 1905 and 1908, a whole bunch of people like Einstein and Sutherland and Smoluchowski and Langevin showed pretty conclusively that it's thermal noise that drives Brownian motion. And I'm, going to just sort of sketch a few essential pieces of that because I know you've seen it before. But from an experimentalist, there's an experimentalist perspective. There's a couple of really important observations when you can do this experiment again with a really fancy microscope and a computer and image processing and all that stuff. That on average, over any given time interval tau, if you average over the entire trajectory, so it could be a time average or it could be an ensemble average across any particles, that there's no net flux of particles to the left or right. So this is a one-dimensional representation. Okay, of course that means in the absence of an external force. So on average, they don't go anywhere, but on average they go somewhere, right? Because the variance of the displacements increases proportional linearly with time with the prefactor we call the diffusion coefficient. Okay, so that's sort of there's no theory required, right? You can just go to the lab and observe that. Okay, so I love the way Einstein did things, but I'll use the Langevin approach, right? So in the Langevin approach, you just write down Newton's second law. You have ma, and then on the right hand side you have the force. And as I'm sure you've seen multiple times, there's two parts you typically have, right? So this is the drag term, and this is the noise term. Okay, and this was the approach that Langevin introduced. And then similarly to the fact that the experiment has such simple statistics for the force, one assumes very simple statistics, very simple statistics for the displacements. Langevin assumed very simple statistics for the force. You assume the time average of the force was equal to 0, and that unlike Ludovic's swimmers, there was no correlation except at the same time for the driving force. So there was absolutely no correlation, and in order to ensure that everything matched equipartition, or more generally equilibrium, the prefactor in the noise famously is twice the drag coefficient from up here and kT, and this is to make equilibrium happy. Okay, so that's the part we know well. And let me just, before I go to the next board, in the upper right-hand corner, just say, because it's going to be relevant to the experiments, I'm going to show you more modern experiments I'll show you in a minute. This in Fourier space, right? So if we Fourier transform it in the time domain, then we can talk about the spectrum of the random noise as a function of the frequency. And that's proportional to the drag and to KT. It's the so-called white noise spectrum, right? At least it's a white noise spectrum for a viscous fluid. It's not a white noise spectrum for things with complex rheology, but we'll get to that in a minute. I want someone to ask me a question by the time I finish erasing this board. Or maybe I'll ask you a question if you don't ask me a question. That might be a good threat. Yes, thank you. Because I didn't have a question. I was trying to think of a question in case nobody asked. Yeah. The higher order correlations? I don't really, no, I do remember actually. So for the Langevin equation with overdamped Langevin equation, so you ignore the left-hand side, the probability distribution is exactly a Gaussian that just grows over time, and so everything is just it's all second moment. There's no higher order stuff. And even if you put it in a constant force, it still actually is all, then it's just first moment and second moment, and you have to start having gradients and stuff of force to get higher order moments. Any other questions? Okay, so, now let's get back to active matter, right? So I just told you that this isn't active, right? But you can do the same experiment inside of an active material, right? So let's talk about Brownian motion inside of a cell. And the first experiments and the theoretical framework were put forward in this really beautiful paper by Lau, Lubensky, and Crocker in 2003. But there's actually there's a more recent paper that I'm going to tell you about. It's from Dave Waits' lab in 2014. Right, okay. So the basic idea of this experiment is that they follow passive particles diffusing in the cell, and they just measure the mean square displacement as a function of time. Nice and simple, okay? So now this is where it gets tricky, because I'm going to start trying to draw data on the board. And I'm going to try to draw data in a way that is sensitive to the scales of the phenomena. I want to get the length scales right. I want to get the time scales right. So it's sensitive to sort of the physical experimental reality of it, but I also want to be sensitive to the theoretical reality, and that I want to get all my exponents right. So anyway, so bear with me. Hopefully we can do this. So we're going to put the mean square displacement as a function of lag time on the vertical axis. We're going to use units. I don't know, have you guys seen units so far? These are real units. This is a micrometer. It's a millionth of a meter. A meter is about this big, just in case. Are there any experimentalists here? Oh, great. Hi, nice to meet you. Yeah, okay. It's great to meet all you too. I'm just kidding. I want to learn from you guys. All right, so we're going to have microns squared on the vertical axis, and on the horizontal axis, we're going to be looking at time in seconds. And like all good experiments, well, sorry, not all good experiments, but all my favorite experiments, let's put it that way, that's a little more generous, is that I'm plotting, these folks plotted it on a logarithmic scale. Okay, right, so this is 100th of a square micron, a 10th of a square micron, and 1 square micron. And similarly, on the horizontal scale, we go from 100th of a second to 10 seconds. How many decades do we have in between? We have 3 decades in between. This is 0.1, and this is 1 second, okay? So, and this data is, let me remind you that there's a passive 100 nanometer sphere that they're following in the cell. They're doing exactly the same experiment that Brown did, except they're putting it inside of a living cell, okay? And what they saw is the following. So, okay, so at short times, it was about 0.01 microns squared, and around a 10th of a second, it turned up, reaching about 10 and 1. Yeah, maybe I can, I want to get the slopes right. Okay, 10 and 1 here. And so this is a log-log plot. This had a slope 1. Okay, so at long time scales, things looked like normal diffusion, but on short time scales, something else was happening, right? And I'm going to label this as WT. Anybody know what WT is? That's sort of biospeak. Yeah. Wild type. That means that it's actually a little generous. So the thought is that it's meant to be a healthy, normal cell. There's nothing healthy and normal about ripping a cell out of an organism and growing it on a piece of glass, okay? But as healthy and normal as it can be in your experiment. That's the wild type, okay? And then what they did is they did something very harsh, okay? They applied a small molecule inhibitor, a chemical compound that prevented the cell from being able to make ATP, okay? And ATP is an essential piece of the metabolism, as was alluded to earlier, and once you remove that, basically the metabolism comes to a screeching halt. And in that case, what did the mean squared displacement look like? It looked like this. So I'm using, green on green is not good. Let's use orange. It was basically flat with the slightest little uptick. And so this is when you remove ATP. This is like the harshest thing you could do. This is completely removing all of the activity from the system at all. You might have thought, you know, that when we removed the activity, that it would have looked like Brownian motion as I described it to you, right? Because there's no activity here. There's no activity here. What's the difference? Yeah, if you look at this MSD, what do you conclude about the medium? It's what? It's high density. It's basically rheologically, you would say it's elastic. If this thing is flat, it has a zero frequency shear modulus. Okay, it's not quite flat. It's sort of, you know, so it suggests that it's pretty glassy. But as we'll see in a minute, I'll tell you, you can't actually directly interpret these terms in terms of rheology, which is really important, and I'll explain that. Okay, so this is completely hitting the cell really hard. There's an intermediate case. What they did, I want to find a different color now. Intermediate case is where they, instead of getting rid of all the metabolic activity, they just got rid of motors, okay? And they got rid of one particular motor called myosin 2, okay? And it's a molecular motor. What that means is it's basically a swimmer, except it needs a track to swim on, okay? And it moves discretely and more stochastically than a swimmer. And when they did that, you could see that there was sort of a drop of about sort of a factor of 3. It still had an exponent of 1, actually. And long times it still ended up flowing. So there's other things in the metabolism other than these myosin motors that contributed to the behavior. But myosin is a big chunk of it, okay? This molecular motor. So the real question is, well, what happened? How can we interpret this, right? So what changed? What changed with or without, let's just say, ATP, just to be concrete, right? There's two things that could have changed when we removed myosin or ATP. What are the two different things that might have changed? Yes, good, yeah. So the first thing is it might have changed either the mechanical properties, I'll say rheology because it's fun to say rheology, okay? It might have changed the mechanical properties of the network, but the other thing it might have done is it might have changed, and in fact it does also, change the noise spectrum, okay? Changes the way that the system is driven, okay? So the question is, which one of those is it? And in the original experiment, when Crocker and Lubensky did it, they basically assumed that it was all about noise and there was no contribution from the rheology. And then the more recent experiment, they didn't assume anything. Well, I'm sure they assumed stuff, but not as much stuff. And they showed quite clearly that the activity both changes not only the noise spectrum, but also the material properties, okay? So, mhm. Yes, I mean that's so that's for me one of the most interesting questions about active matter, right? And a lot of times we talk about one or the other, but as Ludovic was telling us today, right, the question is, does the noise change the glass? Does it change the rheology, right? But here the trick, and you know what we really ultimately want to be able to do is sort of given a noise spectrum, right, or some type of flavor of activity, can we predict what the structure and the properties like the rheology will be? So that's a great question. Other questions? Yeah. No, not at all. I mean, it's actually, you know, and if you looked at a different cell type, you probably wouldn't see that, right? And so, in fact, when I first saw that, I was sort of almost skeptical, no, I was skeptical. So I think one important thing is that, like with, it's real about a biology. It's really, really important to not overgeneralize your results. And so the way I finally, you know, came to understand why this in the long time scales looks diffusive, it really depends where in the cell the particles are living. If they live in a part of the cell where the actin network looked like that actin network I showed you, it would never have done that, right? If they were attached to microtubules, they would have had an exponent of 2 because they'd be moving ballistically, right? So my interpretation is that these must have been in a part of the cytoplasm which was sort of glassy, but not deeply directly connected with the cytoskeleton. And that what determines that is what, you know, what type of particles you use, how big they are, what's on their surface. That determines where they go in the cell, and then that will determine whether or not this is slope 1, 0, or 2. So, but that's a really important question. Yeah. This cell was just like that schematic diagram I showed earlier. So you know, the nucleus of a cell depends on the cell type. It's usually about 10 microns ish, and depending on the type of cell, it could be maybe 20 microns across or a few hundred microns across. This particular cell type, I don't even remember what the cell type is. This is what happens when physicists talk about biology. It's a cell. Okay. All right, so now I'm going to describe Mingguo's experiment to try to answer this question definitively, okay? So we're answering that question, and so there's 2 parts of their analysis, and to sort of for those of you that understand what I'm about to say, basically, you know, they're basically doing fluctuation dissipation tricks inside of the cell to try to see how the noise spectrum changes. So the first step, what they do is they apply a known, well, after they've already done that, they apply some known time-dependent forcing to the same particle inside of the cell. Okay, not just letting the noise spectrum do its own thing, but have a magnitude of a force which is bigger than the noise spectrum. So they have to assume linearity, which might be a totally dodgy assumption, but they had to do it. So they apply a relatively big known force with a known frequency dependence, and they measure how the particle moves. And again, they like to do this in Fourier space. I dropped my paper. Okay. And so the way they measured the force, I'm not going to get into these experimental details, but I'd be happy to explain them to any of you that care. The way they applied the force was with a laser trap, a focused laser beam, an optical tweezer, a tractor beam, whatever you want to call it, and they just moved the focus back and forth at a given amplitude and frequency. And then they measured how much the particle moved. And the idea here is if you measure both this and this, you can measure basically the stiffness of the cell, okay? And that stiffness they call K, it's like a spring constant, little k of nu, which is just the ratio, including phases, we're doing this with complex stuff, of the force over the position in Fourier space, okay? So this is just like you would do rheology normally, but this spring constant depends on the size of the probe particle. You could re-express this in terms of a shear modulus. They didn't do that in this paper primarily because they were trying to publish it, or they did publish it in a biology journal. And if they said modulus, it would have been harder to get it in. Saying spring constant, it's easier. Yeah. Yeah, I'd love to know how they managed all this. My understanding is that they just must have and had to have done a ton of averaging, right? When you're doing experiments with cells, it's what's really tricky about it is the first thing you have to do is you have to decide which cells am I going to consider? Is my cell happy or not? And there's hilarious YouTube videos about happy cells and how biologists decide whether or not a cell is happy. It's hilarious. It's really, really hard. Biology is infinitely harder than physics. It really is. I actually disagree with most people that think that physicists are harder. I mostly think it's just that physics is easy. Biology is super complex, okay? Anyway, so I think it just boils down to very careful selection of the cells, making sure you always use the same place inside of the cell, and then just doing a ton of experiments. That's the only way they could have done it, assuming they did it correctly, and I assume they did it correctly. Okay, so this is what they measured. Okay, so here on this axis is K prime. So this is the real part of the stiffness. It's like G prime, okay? And it has units of Pascal microns, okay? Basically because this modulus is on the scale of Pascal and the particles are on the scale of microns. That's roughly how to think about it. And then we have the frequency on the horizontal scale. And that has units of hertz or inverse seconds. They considered frequencies from about 1 hertz to 100 hertz. And I'm just going to show one value of K prime in the middle of about 10. An important thing I want to say is what they found is that the value of K double prime was much, much less than K prime. Okay, so to keep the plot from getting really messy, I'm just going to show you the K prime part. Because K prime, double prime is much less than K prime, experimentalists would say loosely this thing is more of a solid than it is a liquid. And if we look at the wild type, I'll try to use the same color. If it was really purely elastic system, right, the stiffness would be independent of the frequency. But because the system is probably soft and glassy, it was pretty power law with a slope of 0.15. Okay, so a really, really low frequency dependence. And then, this is the wild type, right? So this is the cell doing its normal thing. And then when orange and red, what I found was super interesting when they either got rid of ATP, or got rid of the molecular motors, they basically saw exactly the same thing. Okay, so the interpretation there that tells us that the main element of the activity that affects the material properties of this particular cytoplasm is the myosin, right? So myosin, this molecular motor, really is the dominant factor in determining the properties of the system. Okay, then the next thing that they did, right, is to measure. They took their same trajectories from before, and they sort of calculated x of nu, for the unknown case. For unknown f of nu, that's sort of the native noise inside of the cell, and they basically, used basically the same sort of formula as before. At some level, it's exactly the same. We have force is proportional to a stiffness times a displacement, right? But now, because this is Fourier transformed and it's like it's sort of ensemble average, right? It looks more like a correlation response function that you would talk about in non-equilibrium stat mech. Yeah. Okay, so so F and X are complex numbers. K is complex. K prime is the real part. So if it's And here I'm saying that the part in phase with the force is dominant over the part that's out of phase, and so that's elastic. It looks like I'm more like a spring than it does like a dash pot. Okay. Good. Okay. So the cool thing is now that they've measured, they have an independent measure of the mechanical properties, and separately they measured this, they can actually calculate the noise spectrum inside of the cell, which I think is great. Super cool, but not super cooled. Okay, so this is what the noise spectrum looks like. So frequency on the horizontal axis, and on the vertical axis we have the power spectral density of the force. It has units of newtons squared seconds. The second, so the F squared is, it's clearly just force squared. The second is here because it's a power spectral density. It's really newton squared per hertz, right? And that's why it's newton squared second. That's the experimental detail that's worth knowing. Yes? They're assuming that it is. Yeah. So I mean, but they haven't changed. The noise is the same in both cases. So this is what's really nice about their measurement over Lau and Rubensky and Crocker. So and Lau, Lubensky, and Crocker, I don't remember exactly what they did. So I don't want to criticize that. But the key thing here, right, is that this measurement is done in the presence of the native noise spectrum of the cell. So whatever properties the cytoplasm has due to its own noise spectrum, it was there when this happened. The only reason that they were able to measure this, right, is because they did lots of ensemble averaging, right? Or they had they limited themselves to certain frequency ranges, and they needed to make sure the force was big enough to overcome the native force levels, which calls into question whether or not it's really linear. But let's just, this is better than anybody else has done. So like we can criticize it, but really it's a tour de force. Yeah. Yeah, exactly. Yeah, I'm just like, yeah, exactly. And that may be problematic or not. And I didn't read all the supplement. I don't know if they checked. Like when you're doing good rheology, you make sure in their linear regime. I don't know if they did that. So, okay. All right, so let's do the dead case first. Oh no, I have to label my axes, I'm not done. Again, we go from 1 hertz to 100 hertz with 10 in between. And again, I'm just gonna give you one number because you can figure out the rest. This is about 10 to the minus 28 over here, Newton squared seconds. So, that sort of suggests if you put in a time scale of about 1, right, that suggests that the scale of the force is like tens of femtonewtons, if that means anything to you, okay? But first, let's look at the case of the cell where we've removed ATP, where the noise spectrum should be thermal. And what color was that? That was orange. Where's orange? Here's orange. When the noise spectrum was thermal, they saw something like this. It was a straight line minus, this is without ATP, and the line had a slope of minus 0.85, right? So it had power law, okay? Amazingly, this is exactly what you would expect for a thermal system with no activity and this rheology. Okay, so quantitatively, when they get rid of ATP, they observed and they further assert that it has the right rheology. So this suggests that perhaps wait, that's confusing. No, this was also 0.15, and because they used. See, this is what's so great about the experiment, right? This one was also 0.15, and they used the stiffness that they measured in the absence of ATP. So this is really the correct noise spectrum. I mean, the correct noise spectrum, and it was perfectly consistent with a thermal noise spectrum. Okay, then they did it for the wild type case, and wild type is yellow, and sort of looks like this. Out here it was -2, they had some explanation for why it was -2, which I didn't buy. And the key thing here is that at high frequencies, the noise spectra converged onto each other. And this is actually a really big idea that a lot of people have been talking about. And I don't know if it's trivial or profound or even correct, but biophysicists assume it all the time, that at high frequencies, the noise spectrum of the active system has to approach that of the thermal system. I've seen it asserted, I've seen it measured here, but I don't really know how generic that is. Yeah, so I think, I don't really know why. Well, you could say they say that because they saw it here, but when they don't measure it and they just assume it, they say, okay, molecular motors and whatever physics they say that drives the noise spectrum have some characteristic timescale. And if you're measuring noise at timescales that are much faster than any motor behavior, you must just be seeing the molecular vibrations. Seems very reasonable, but I just don't know how robust. I don't know how robust, and I don't know when you can really get away with it. I bet someone could. I don't know if someone's done theory on it. If not, maybe one of you can. And finally, here's the Myosin case. The Myosin case was, that was supposed to be orange. No, that wasn't. Myosin was supposed to be red. Myosin was supposed to be red. Myosin looked very similar as -2, and overall is about a decade below the wild type. Myosin 2, or minus myosin. And so what this says here, actually, which I think is really cool, is that the myosin did affect the noise spectrum, right? But not nearly as much as everything else did, right? Especially as you go to lower and lower frequencies, other contributions to the noise completely dominate over myosin. So I think there's an interesting point here is that sort of, I would say myosin 2, the activity associated with myosin 2 sort of is more strongly couples to the mechanical properties. It's really changing the structure of the material, where all the rest of the metabolic processes of the cell captured from ATP, where the ATP dependent processes, all the rest of them are dominating the noise spectrum. And for me, this is like, I have personal reasons for getting excited about this, because I had a paper, which you mentioned, where I claimed that ATP was the important thing. But anyway, but nevertheless, they still presented a model in their paper saying that the noise was all about myosin activity. So, which, maybe describes some of it, but doesn't describe most of it. So I think there's another great opportunity here for thinking about, how generic, and what's great about killing out ATP is this really generic biochemical activity. It includes processive motion. It includes swimmers. What were your three things? Swimmers, and then shape changes. All those pieces are all dependent of ATP, and this sort of shuts them all down. Yeah. Yeah, it depends on the cell type. Okay, so how much time do we have? Okay, so we have this paper which I was going to tell you about, but I decided not to. But I'll tell you a little bit about it now. It was in bacteria, and we looked at the Brownian motion of passive particles inside of bacteria, and we found a big drop in the mean squared displacement of passive particles inside of bacteria. And what was really interesting about that result, and why we got so excited about it, was because initially the thing that got me most excited about it is no one's ever identified a molecular motor inside of a bacterium, a canonical molecular motor. And so I was pushed to make this argument that the sort of generic ATP activity can drive enhanced by Brownian motion. And the example, the mechanism that I like the best has been already mentioned today by Ludovic and has been alluded to by some of Ramin's papers, is the idea of conformational changes of proteins, right? So the way I think about it, okay, is that any conformational change of a protein that is driven by ATP hydrolysis liberates free energy. If it liberates free energy, it can do work on its surroundings. So every ATP-driven conformational change is a source of mechanical noise in the cell, and that's my hypothesis for what this is. Because if you calculate sort of all the thermal stuff, and we're actually going to do that in the later part of our lectures in a few days, we're not heating the system up, right? These systems are small. heat diffuses quickly. So my guess is that it's conformational changes. The other thing, which was almost on your list but not quite, is not processive motion of individual particles. I think another really important to the mechanical nose, I'm just hypothesizing here, inside of cells, especially bacteria, is just polymerization forces, right? So you have processive motion inside of a cell. Every time you replicate DNA, you're squirting out a polymer in one particular direction, right? And that generates shear forces that can mix things up, making, either replicating DNA or making RNA or making protein wherever you are. I think all those things, quite broadly can drive differences to the noise spectrum. But what's really interesting is that they don't all couple to the material properties, right? So maybe, what do you need to couple to the material properties? And in the paper of ours, which I'll just write the author since I'm not going to tell you about it. It's Brad Perry and me and a bunch of other people, primarily Christine Jacobs-Wagner, who was the biologist driving it. There we actually concluded that it wasn't a change in the noise spectrum. We concluded it was a difference in the mechanical properties. We argued that this metabolic activity fluidized the glassy cytoplasm. Okay, that's what we argued. But because we weren't able to do this measurement, we couldn't separate the noise from the properties. We had nothing, we couldn't, that, and at some level that is not a justified conclusion. But it's a fun conclusion to think about, as Ludovic has shown. Questions? Yeah. Yeah, so on the last, this is their conclusion that this is thermal noise. This is they got out of ATP hydrolysis, and their claim was that this noise spectrum was consistent with this rheology, and they plugged in KT, and supposedly all worked. Yeah. Good question. Yeah, right, because this is a power law, and this is a power law. So I would have expected that to be a power law, but it doesn't look like a power law in their plots. Maybe it looks more like a power law in their plots than in my drawing, so you should go read the paper. But I think it looked pretty, to me in the paper, it looked pretty darn flat and then went up a little bit at the end. It didn't look. This one? I don't know exactly what the slope should be, but it should be subdiffusive. It should be a really, I would expect it to be a really, really small exponent, maybe 0.15. Maybe. By something really small, because I mean, if it was purely solid, it would have slope 0. If it was purely viscous, it would have slope 1. If it was mostly solid, it's power law fluid, should be a little power law, it's, close to 0. Closer to 0 than to 1. Okay, all right. Okay, so we have 10 minutes. Let me just decide what to talk about in 10 minutes. Yeah, so I think it's, maybe I'll just set it up in this today, and then tomorrow I'll finish it. I want to talk a little bit about, because I think it's really fascinating, about how myosin activity can have a dramatic change in the mechanical properties of the cell. And there's a really long history of beautiful work done by people like Dave Waits and Fred McIntosh looking at that, and I'm going to I want to summarize, I want to show you some of the key results of that to understand the origins of, how that special type of activity couples to the mechanical properties of polymer networks. And I think today I'll probably just get to the point of sort of setting it up, and then tomorrow I'll give you the results. Okay. So, yeah, so the key point here is here's a concrete example in a simplified system of how activity modifies material properties. And we're going to consider the specific case of disordered actin networks. One type of an active gel, okay? All right, so actin is a polymer. Actin is a protein that polymerizes to make relatively rigid filaments. And I showed you some pictures. I drew a sketch another sketch earlier of an actin network. But the important thing about when you draw an actin network is that a so-called semi-flexible network, semi-flexible polymer network, is that the persistence length of the chain is comparable to the distance between the crosslinks. So unlike rubber, where between the crosslinks it would be all loopy and fractally and crazy, and a semi-flexible polymer network, it's got, you know, relatively modest fluctuations. Okay, and so in the sorts of networks, and so what we're going to do now is we're going to talk about some in vitro experiments done again in Dave Waits' lab. He was my postdoc advisor, so I talk about him all the time. Okay, so anyway, so in vitro experiments done in Dave Waits' lab to sort of understand this question of how myosin changes the stiffness of networks inside of cells. And what they did is they took actin and they cross-linked it in two different manners. They cross-linked it passively. Okay, so these little things are passive crosslinks. And depending on the experiment, they either used alpha-actinin or filamin A, I think it was. Those details don't matter unless you care about biology. But the point is that these act like sort of traditional crosslinks that you would think about in a gel or a cross-linked elastomer. They don't do anything. But then they have a second type of crosslink, which I'm going to show very schematically, like this, the red ones with little arrows. Right? And these little arrows, I mean the active ones, the active crosslinks are myosin 2 bundles. Again, if you aren't interested in biology, that name doesn't matter. What you really care about is the way I drew it. The point is that this thing generates a little force dipole. Okay, it not only cross-links the two trains, it doesn't just passively cross-link two strands of the polymer network. It pulls on them, right? It pulls on them, dynamically generating force. And so if you look at the rheology of these things, okay, so these networks are elastic. Okay, so and what I mean by elastic is at zero frequency. Well, actually, maybe they're not quite at zero frequency, but down to relatively small finite frequencies, they have a finite shear modulus. Yeah. They potentially have an on rate and an off rate, but that the binding and unbinding obeys detailed balance, right? So it's reversible, right? Whereas here, the binding, pulling, unbinding, binding, pulling, unbinding involves cyclical hydrolysis of ATP to ADP, creating one of those cycles that I was showing you. That cycle produces entropy, irreversible, generates force, right? So so yeah, it's really, sometimes it's hard to differentiate these things, but that's, I think, the right way to explain the difference. Okay, so these things are basically elastic, or at least viscoelastic. And I mentioned that these things act as force dipoles. Actually, the mechanism by which these force dipoles work is highly non-trivial. People for decades assumed that they understood it, and then someone named Martin Lenz and Aaron Dinner showed everybody that they got it completely wrong in a beautiful PRL from 2012 that I really like, if you want to read that. Anyway, the actual mechanical mechanism of how these things work is highly non-trivial and interesting. And a couple of facts that I want to say about them is that, so the networks are elastic, and that the myosin activity drives contraction. And that, and you can see that in two different ways, sort of in an unconstrained system. In a paper by Bendix and Waits, In 2008, they saw a 64 times volume reduction. That as ATP was hydrolysis, they cross-link the thing, they turn on the ATP, they provide ATP. As it contracts, it has a tremendous volume reduction, okay? So these things really contract. The other thing they do, if you restrain them, they generate force. They can do work. They can do work on the outside surroundings, and they have this really cute example where they took 2 oil drops that were stuck to a cover slip, and then they polymerized a network of this stuff with both of the crosslinks between the oil drops, and then when they added ATP, what you clearly see beautifully is that these oil drops get deformed. They change their shape, and the network contracts and modifies itself, right? So here it's clear, because it's deforming an outside object, you can see and you can calculate, because it's just a liquid droplet, exactly how much force it generates, okay? So, yeah, so those are, I think, the simple facts that I want to tell you about the network and its properties, and then tomorrow I'll tell you about some more quantitative things, but I have time for a couple of questions before we go. Yeah. Absolutely. So this is one, I mean, this is just, yes, this is not a universal number. I used under some set of conditions, they saw this. My point is that it can be very big. I consider it to be very big. Perhaps it could be much bigger, and in other conditions it would be 0, right? Yeah. Yeah, so there's so notice how sometimes I just said myosin, other times I said myosin 2. The full name of this particular myosin is non-muscle myosin 2. So there's a whole bunch of different myosins. Some of them walk so-called processively. That means in the language of Ludovic, the tau is really big. But in other myosins, the non-processive ones, they have a small tau, and they basically walk a little bit, fall off, go back on, walk a little bit. They don't have, the Piclet number is smaller. Okay? Let's have dinner.